All, this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. The discussion today is about the Oxford's vaccine, and the the trial has been halted temporarily, hopefully, and then it would be resumed. And there is an independent committee of experts that is looking into when to start the trial again. So I wanted to make sure that we can look at what happened. And in this process, today's takeaway are a couple of things. Number one, what is going on with Oxford vaccine? And number two, can a vaccine cause what we are seeing? Because the discussion that we were given, or the message that we are given, is that it is possible that the vaccine caused this issue, or it is also possible that the person already had the issue and then the vaccine just simply was there when that diagnosis is made. So I wanted to make sure that we can look at if it is possible for a vaccine to contribute to create neurological symptoms, and if that is the case, how would that be done? And this discussion that we'll do today, it is the same for the viruses as well. A vaccine, whatever is the mechanism that we'll discuss, it is a similar mechanism as a virus can do this too. So viruses or bacteria or fungi or parasites can also give rise to autoimmune diseases, just like vaccines may be able to do that too. So with this, let's start our discussion. I hope that from our last lecture with thiamine, many of us have the cool beans have started taking some thiamine as well, which is important. So there is a question about leronlimab. So this particular discussion today is about the Oxford's AstraZeneca's vaccine that is for the COVID-19. So let's start. So once again, welcome to everyone. Here is the vaccines discussion. So we are talking about, this is the clinical trial. This trial is for, if you see here, phase 3 double-blind placebo-controlled study of AZD1222. So I'm reading here at the top. The AZD1222 is the vaccine that was originally called ChAdOx1 from Oxford. So then they went into a partnership with AstraZeneca, and then they're making that vaccine together, and that is what this vaccine is called. And I'm hoping that I am pronouncing AstraZeneca correctly. So if you see here, the vaccine is in phase 3 trial. I think this is for the US. The participants were supposed to be 30,000 in the US, out of which I believe 20,000 would have received the vaccine and 10,000 would have received a placebo, and then the outcomes would have been seen. So this is the discussion. The trial has been stopped for a second. So I'm going to show you a few things here. All of these links are present in the description. Here is a link which gives us a little more description of the vaccine itself. Then this is the link to how various scientists and experts are reacting to the stoppage or halting of the vaccine from Oxford. Here is stat talking about what really happened. So I think that this is the most useful one to look at, where the CEO of the AstraZeneca is talking about what was the patient, who was the patient, where are they from, and what is the possible issue that they're having. So with this, what I have done is, in today's discussion, we are also going to talk about transverse myelitis, because that is the disease possibly that was seen in one of the patients or one of the participants. We will also talk about multiple sclerosis, because these are kind of overlapping diseases. We will also talk about blood-brain barrier and how immune cells can actually end up in the blood, in the brain, and cause damage or inflammation. And we will also talk about the brain cells that are responsible for the myelin sheath, which I'll explain what it is. So we'll look at the mechanism of the damage as well, and we'll look at the immune system's behavior too. So if you just wanted to have a quick synopsis of the discussion, the synopsis is that there is one serious event in a participant from UK who is receiving the vaccine. So the Oxford's vaccine has been halted for the temporarily, and then we would start again. I'm going to explain how that is possible that this can happen. So let's start. So here. So first of all, the vaccine itself. So the vaccine originally was called CHADOX1, CHADOX1. That was from chimpanzee adenovirus Oxford vaccine 1 for COVID-19. So because this was an adenovirus that is of chimpanzees disease, and then it was modified to put in the coronavirus, SARS-CoV-2's spike protein genetic material in it, and that modified and weakened virus is the part of the vaccine. This vaccine is now called AZD1222. Vaccine has been trialed in UK, Brazil, South Africa, and now since August in United States. So the important thing is that this adenovirus that is in the vaccine cannot grow in the human. So all it is doing is it is bringing the RNA or the genetic material into our cells, but it cannot further replicate. And what it brings to our cells is the genetic material for the SARS-CoV-2 S protein or spike protein, against which we are trying to train our immune system cells, so that when the actual virus comes in, arrives in our body, our immune system cell can recognize the S protein and bind with it and neutralize the virus and kill it early. So now the phase was third phase, 30,000 participants in the US were supposed to be there. Dose was 2 intramuscular doses 4 weeks apart. So what is the exact dose? If you see here, they have talked about the dose, 5 multiplied by 10 raised to the power 10 VP. So whatever that finally translates into, not very important for us. Now, what really happened? What happened was that one of the participants from UK, she developed transverse myelitis. That is the adverse event, serious adverse event. And because of this, the trial is halted. She is improving. And by the Wednesday, which is, I think, this Wednesday, which is just past, by this Wednesday, she was expected to be discharged after recovering from transverse myelitis. There is an independent experts committee that is now going to determine when to lift the halt and resume the production or the trials. And by the way, there's a lot of money that has been given to Oxford and AstraZeneca for this one. I believe $1 billion from the US. Other countries have given money. The production of the vaccine is already on its way for UK and I believe for US. So it is not something that that was just 1,000 or 2,000 vials that were there. They are in the process of production and they are halting, as they say, temporarily. So we'll see. Now, when we talk about transfer, so this is it. This is the basic discussion. I'm going to now explain the mechanism of why this happened. And can this happen with other viruses and vaccines? So if you want to see the mechanism, please stick around. So here is a section of spinal cord that I have. And in this section, what you're seeing is you're seeing a couple of things. One is this area of the spinal cord. This is all called white matter. What does that mean is that our spinal cord, and that is true for our brain as well, it has two kinds of tissues in it. There are many types of cells in there, but two primary kind of nervous tissue. One is the cell body that is actually doing the processing of the things. So that is called gray matter, because when we look at it under the microscope, it looks gray. And then there are the nerve fibers that emerge from the cell bodies, and they go all the way to the other places. For example, from our brain, a cell that is present in the brain would give rise to a nerve fiber that can go all the way to our toes and move the muscles there, or a cell from all the way from the toes, skin of the toe, can come back all the way to the brain to bring the sensation back. So these are long threads. These long threads are coated with a myelin sheath on them, with a plastic-like thing on them that increases the travel speed of the nerve impulse. And we'll talk about that. Here, these white areas are where the threads, the pathways, the tracts are going through. And this gray area that you're seeing, that is where the nerve bodies are present. And for the medicos in here, this is the frontal or the anterior root. This is the motor root from where the fibers come out that will go to the muscles. And this is the dorsal root or the sensory root through which from the skin and the muscle spindles and the Golgi tendon apparatus and all other sensory systems, they bring the information back from our body into our spinal cord and then all the way up to the brain. So this is the sensory system. And for further excellent medicos point, this posterior root neuron or the cell here is called a pseudo unipolar cell. And pseudo means fake, unipolar means it is single poled cell. And why do we say that? Because if we look at it under a microscope, the cell looks like it has a cell body and it has one pole. This is one pole. So we call it unipolar. But basically, these are two poles that have come so close to join almost with each other. So they are actually two poles fused together to look like one pole histologically. This is why we call them pseudo unipolar, fake unipolar. Anyways, that's just a medical student and medical's point here. So continuing on. Now let's see, we're gonna now discuss how the transverse myelitis occurred, what happened, and how did the vaccine participate in it. So first, let's focus on this part. This is a neuron. This could be a neuron in the brain, or it could be a neuron in spinal cord. What it has is, this is the cell body of the neuron. There are dendrites that are the fibers through which a neuron receives signals from the other neurons. And then it has a long thread that goes out towards maybe a muscle or maybe a gland or maybe coming from a skin. So this long fiber is called a thread, an axon. And this can be multiple feet long. So this is a neuron. The neuron that are conducting fast, they need to have the nerve impulse in them, the electrical activity in them to run fast in them. And by the way, I love nervous system. Nervous system is my beloved topic in the whole medicine. So some people think that I really love immunology. That I do. I love cardiovascular system as well, but nervous system is something that I am so passionate about. So back here, these long threads that bring impulses, let's say from the brain all the way to the muscles of the foot, they are coated with a lipid membrane, a waxy membrane, just like the covering on a electrical wire. And this waxy lipid membrane is called a myelin sheath. The benefit of this is that the nerve impulse then travels from one gap to the other gap, and then from the other gap to the next, and then from the next, and then to the end. So it jumps. We call it jumping way of nerve conduction, and it is very fast conduction. So now imagine if for some reason this myelin sheath over here, if this got damaged. So let's say the myelin sheath here became damaged and it was disrupted. So this much of an area of the myelin sheath here got disrupted. If that would happen, then the nerve impulse cannot travel by jumping anymore, because from one node is this area where the nerve impulse is, and this is a node, and this is a node, and this is a node. From one node to the next node, the waxy low capacitance thing, the myelin sheath has become destroyed. So the nerve impulse now has to walk through this instead of jumping or running. And so that makes the nerve impulse conduction slow, which in turn causes sensory and motor deficits and abnormalities, which we see in, for example, multiple sclerosis. So in the nervous system, both in the brain and in the spinal cord, there are other cells as well, other than the neuron. So there are many kinds of other cells. There are some that are connective tissue that are supporting the other cells. There are some cells that are acting as the immune system of the brain. There are some cells that are sitting there and making the fluids of the brain, the cerebrospinal fluid. There are some cells that are sitting there and making myelin sheath itself, and so on. These cells all collectively are called glial cells. So our brain tissue can be divided into two types of cells: nervous system cells that are doing the function of the brain and spinal cord, and then glial cells, which are all other cells present in there. Now, what are these glial cells going to do? So if you look at it, this little glial cell that I made, it has a feeder here. So some glial cells function is to feed the neurons, bring them nutrients and bring them oxygen. Then if you see here, this is a coronavirus that this glial cell has eaten up. So these glial cells can act as immune cells as well. Remember, our immune cells outside are not allowed to go in the brain. So brain has its own immune mechanism. And so these are, for example, microglia are the cells that are present which can work with as immune system cells. So here, this is a coronavirus that the immune system cell of the brain has eaten up. So that is defense against the pathogens. Then these cells can provide support. So if you see here, this little tentacle, this long arm comes in and it is supporting a neuron. So they allow the neurons to be tightly bound in their area instead of moving around as we move. So we provide support and we provide anchoring of the nervous system tissue. So we make myelin as well. So there are cells called astrocytes, for example. Oligodendroglia, those cells make myelin sheath, this waxy layer. In the nervous system, in the peripheral nervous system, that means spinal cord and the other nerves that are out in the body, there are cells called Schwann cells that are responsible for making that waxy coating that goes on top of the cells. So various kind of glial cells: astrocytes, oligodendrocytes, they make myelin sheath; Schwann cells, they make myelin sheath; ependymal cells, for example, they make cerebrospinal fluid. In our discussion, please remember two of the cells today: oligodendrocytes and Schwann cells. They are the one that make myelin sheath, and that is what possibly this vaccine caused a destruction of. So let's see what happened. So here now, from the nervous system, we are going to switch to a concept of immunity, and that concept is the molecular mimicry or cross-reactivity. So if you look at it, let's focus on this side first, this side. Imagine this is a coronavirus. And today I made the coronavirus slightly different. I usually make that with RNA and eyes and everything. But here we are looking at the coronavirus from outside, and we are looking at its surface. Imagine it is a ball, and we are looking at that ball. The ball, excuse me, or the virus has lots of proteins on its surface. And proteins are nothing but amino acids. Amino acids are the bricks in our body, or just like alphabets in English language or other languages. And these alphabets are put together to make structures of sentences or words. Similarly, protein or amino acids are put together to make protein structures. So imagine every single letter here is an amino acid. It is a brick to make a protein. And if you see here, there are various patterns in the bricks. The proteins have various amino acid patterns that make up the protein. So I would like us to look at one pattern here. So let's say AABBC. I just made up a random pattern. This is similar thing as when you sometimes look at the sky and there are clouds out there, and you can look at various shapes in the clouds. So you say, all right, I can see a face there, or I can see a goat there, or I can see a horse there, or a unicorn. There is no shape really in there, but just the pattern of that cloud can allow you to cross-react and think about another shape, another thing. That is the same thing over here. So imagine if this AABBC is a pattern of an amino acid, and here is our astrocyte. Astrocyte, or let's say more oligodendrocyte. So I should not call it astrocyte. oligodendrocyte. So oligodendrocyte is here. Dendrocyte or Schwann cell, the myelin making cells. Imagine now that on the surface of this cell as well, I have made the similar pattern here. AAA-BBC. Is it possible to have similar patterns? Yes. We are all, everything organic is made up of tiny amino acid proteins, parts. And so just like all the literature in the world for, let's say, English language is made up of various combinations of those 29 alphabets. I hope there are 29 or are there 26, whichever number of the alphabets. So you can find those patterns in every article that you see. You would find combination of letters that you can recognize. And that is what we do. We recognize them and we say, okay, this is this word written here. Similar is the case here, that here, this pattern and this pattern are matching. Now imagine that our body has an immunoglobulin. Our B cells produced an immunoglobulin, or our T cells have a receptor to bind with antigen, and they are going to bind with this area, AAA-BBC. So this is the immunoglobulin binding area, and it is going to bind here. So good, this immunoglobulin is a nice immunoglobulin. It is going to bind to coronavirus and try to kill it. On the other hand, this pattern unfortunately is seen on our oligodendrocyte as well, which makes the myelin sheath, and this immunoglobulin is going to attack this cell as well because the same pattern is present there. It's like looking at a horse in a cloud. And imagine if you are, let's say, not a horse, let's say something bad that you don't like. So you look at that thing in real life and you say, I don't like it. And then you look at cloud and you see the same shape there and you say, I don't like that shape either. And you attack that cloud. Same is the problem here. This is called molecular mimicry or cross reactivity, where one set of molecules present in one pathogen are going to look like another set of molecules present on our self proteins, and the immune system would attack them by mistake. So when this attack will occur, our cells are going to start dying. So for example, in the case of a vaccine, it is possible that the vaccine is made to attack, let's say, this part of the pathogen. But that exact part is also seen in some of our cells. And if that is the case, the immunoglobulins that the vaccine would make, or T helper cells that the vaccine will make, these would go and bind to our own cells and react against them and cause damage. This is the basic mechanism of how a vaccine or a virus, imagine this was not a vaccine, this was the virus itself, and that also had the same pattern. And we make immunoglobulin against that pattern, and that pattern, immunoglobulin, is going to now attack our cells as well. This is called self-cross reactivity or self-intolerance. Our cell forgot to tolerate our own system because they got triggered by a virus or a bacteria, and they were, earlier they did not react to our own self, but now they saw that pattern on a virus and they forgot for a second that we needed to ignore this pattern, and now they're reacting to the virus and they're reacting to our own tissue. That's what's happening here. So I hope that this part is clear. I hope that this much is clear, that how cross reactivity occurs. So now let's see one more final thing. And that is a question that has been asked about multiple sclerosis patients. What happens in MS or multiple sclerosis or in transverse myelitis? What is happening is, let's talk about transverse myelitis first. If I go back here for this spinal cord piece for a second, here, this one, transverse myelitis, transverse means the whole section of the spinal cord, one area of the spinal cord, both sides, all sides. Secondly, myelitis, transverse myelitis, means that the whole section, including gray matter and white matter, has become inflamed. Why is it inflamed? Why is it swollen and damaged? Because there is our immune system that has started to attack that. Why is it attacking this area? Because immune system thought that it is primed to do this by a vaccine or an actual virus, and in that priming, in that recognition of the epitope is that piece of genetic uh, an antigen that it attacks. Now the immune system is attacking our site. So this is transverse myelitis. If there is inflammation only of the white matter and not of the gray matter, then it is called leukomyelitis. And if this myelin sheath starts becoming damaged, then that would be called multiple sclerosis. Usually starts from the optic nerve and so on. So these are various terms here. But at the end of the day, what happens is, once we have developed this mechanism, the question is, are immune cells in the body, let's say the vaccine was given in a muscle, and the immune cell outside of the nervous system have become primed and have become trained to attack a specific antigen. Why did they enter our brain or spinal cord? They're not allowed to go there because we have a blood-brain barrier. And there is a study here. I have linked that study here. This is a study how rogue immune cells cross the blood-brain barrier to cause multiple sclerosis. Very, very fascinating. So here is what happens. Scientists found that T helper 1 cells, remember T helper 1 cells? These are the ones that would then go and make cytotoxic T cells active and cause the damage. T helper 1 and T helper 17. Remember, we talked about T helper 17 yesterday, that they are T regulatory cells, and sometimes they can become dysregulated and they can cause actually more inflammation than trying to suppress it. They have seen that T helper 1 and T helper 17 cells actually cross the blood-brain barrier that they're not supposed to cross and go into the brain tissue and cause the inflammation. So they wanted to understand how does this happen. So they found 2 mechanisms. And luckily, in the inflammation part 2, we discussed this as well. Remember, we discussed that the cells can get out of the blood vessels by 2 mechanisms, transcellular passage and paracellular passage. So I'm going to very quickly show it. Here is the transcellular. This is a T helper 1 cell. It attaches to the endothelial cell of a blood vessel. This is an endothelial cell. And in the endothelial cell, there are small pits, there are small caves on the surface of the cell. And those caves are called caveoli. So on that caveoli, the T helper 1 cell attaches. So now one cell is attaching to another cell, endothelial cell. And with this caveoli, it allows the cell, the caveoli allows the cell to pass through the cytoplasm like a boat and go all the way to the other side. This is called transcellular passaging. And so one cell pierces through the other cell and gets to the other side. So here is a T helper 1 cell that has come into the brain tissue by passing through the endothelial cell of the blood vessel. The other thing that they found was the T helper 17 cell, which is a T regulatory cell, that has found its own mechanism to become a rogue cell. What it does is it opens up the intracellular junctions, the junctions between the 2 cells. It can open them up and then pass through them and go into the brain. That is so clever of that silly cell. So here is the T helper 2 cell, 17 cell. It passes through this and goes in as well. Once these cells are inside the brain's privileged area, they would then go and attack all kinds of cells in the brain. And when they would attack the oligo, I keep writing this as astrocyte, which is not correct. So glial cells or oligodendrocytes or Schwann cells, when they go, these are the myelin making cells. So when these immune cells have come in to the brain, now they're going to attack these cells and cause their damage. That damage would cause inflammation in the brain, and it would cause demyelination or destruction of the myelin sheath, which would then, this is like take an electrical wire and remove the plastic coating from it. And the result is now that wire is not going to conduct correctly. In our real life, the wires would continue to conduct. They would just cause electrical currents for others, or they'll become hazardous. But in a brain, when you take the coating off, the wire stops working correctly. And that causes all of the issues that we see with multiple sclerosis or transverse myelitis or leukomyelitis. This is the basic pathology. And this is what happened in the case of that patient. Fortunately, in majority of the cases, this is a reversible condition. And so hopefully she has now been discharged and she has recovered and she is okay and the inflammation is gone. So if that is the case, afterwards, they would start the process of trial once more. But this is what was observed. This is what we are seeing. So after this, I wanted to also discuss about one more thing. And let me just very quickly show you the article. It is an interesting article in Yahoo here. And that is, dentists are seeing an epidemic of cracked teeth. What's going on? And here is a dentist in New York who said, I've seen more tooth fractures in the last 6 weeks than in the previous 6 years. And the reason, so again, this is an anecdote, it is not a study. But the question, I wanted to discuss this with us all cool beans over here, because this is important for our health. What is happening in this case is that they are speculating that we have started working from home. And when we work from home, if I can, actually, I'm going to stop sharing my screen so I can show you my posture. So imagine that this is how I'm sitting and I'm working. So we kind of are looking at our cell phones slouching like this, or we are looking at the TV all the time sitting like this, or we sit in a C shape most of the time at home on makeshift offices at home. That causes a stress on the spinal cord and the spinal vertebrae, and that compresses our nerves. And if you would like to understand more, I can do a complete topic about how this, how are the nerves that are coming out of here and how do they get compressed. So we can talk about it. But at this time, if you just see that if we are sitting like this, that causes the nerves to be compressed. So now the nerves that are compressed, that are going to our temporomandibular joint, these nerves are irritated because of compression. And they, when we are sleeping, can malfunction and cause us to grind our teeth. And grinding the teeth can then cause fractures as well. So that is one possibility. Another possibility is stress. So we are continuously in stress for months now. And the stress, unfortunately, the media loves to actually accentuate that stress. They love to run cycles, news cycles that actually make us even more scared. And then they probably get more views. But the point is that whenever we become scared, our sympathetic nervous system fires. Sympathetic nervous system is a nervous system that is helping us. So that, imagine when you are angry. When you're angry, your teeth are clenched and you're kind of angry and you're all, toned up. So when you are upset and you're stressed, you clench your teeth, and that happens during the sleeping as well. And the teeth grinding occurs, which can then fracture the teeth. Nowadays, so much of the grinding occurs. So this is another possibility. The end result of this is, and they gave a very interesting little way to look at it. The dentist said, observe yourself that are your teeth touching while you're doing normal activities? If they are touching, that means you are stressed, and your teeth normally should not touch if you are not in a stressful state. So they said, if you are touching your teeth normally nowadays, then please realize that you may be grinding your teeth as well, which means you are under stress, which means your posture may be wrong, which means your teeth, not only they can be fractured, but that means your rest of the immune system and your rest of the health is also not optimal. So for that, please make sure that you do your exercises. Please make sure to figure out ways to stay happy as well. Maybe some of these lectures that we offer here, and we're trying to discuss at how we're going to get out of here, maybe some other things, but make sure that you are relaxed and make sure that you are able to process this information in a more thought out and less scared way compared to the way media has been portraying. So these are the couple of topics I wanted to discuss. And we'll continue tomorrow. We have the open forum. I know that I received a few complaints that in open forum nowadays we talk more about the Twitter questions and then less about the questions in the live stream. So what I'm going to try to do tomorrow is one question from Twitter and one from the live stream and kind of mix them. It does allow me to prepare better if I have a question already posted compared to a question which is in the live stream. But I'll try to do justice to both of them. If not, then on Saturday, we'll also have one more open forum, and in that, we'll only answer the questions from the live stream. So this is the discussion. Thank you very much for joining me. Please make sure that you can share this. You can please like, subscribe, hit that bell as well, and share this as well. So thank you very much, and I'll see you tomorrow.